What if I told you that some parasites don't just live inside their hosts, they control them? Deep inside the silent darkness of Irish caves, scientists have discovered something extraordinary, a fungus capable of altering the behavior of spiders before killing them. How does it do this? How can an organism without a brain manipulate the nervous system of another living creature? Today, we're diving into one of the most fascinating and unsettling examples of evolutionary biology: host behavioral manipulation. Let's begin. In natural ecosystems, relationships between living organisms are often far more complex than they appear at first glance. One of the most extraordinary of these relationships involves parasites that not only feed on their hosts, but are also capable of altering their behavior. This type of interaction, known as host behavioral manipulation, represents a highly advanced outcome of evolution at the molecular and physiological level, where the survival of one species depends on its ability to directly or indirectly influence the nervous system of another. In recent years, scientists encountered a new example of this phenomenon among cave-dwelling spiders in Ireland. Within the dark, humid, and relatively stable environments of caves and abandoned underground tunnels, researchers observed spiders positioned in unusual locations, suspended from ceilings or clinging to exposed walls, completely motionless and unresponsive. Initial observations indicated that this posture did not align with the natural behavioral patterns of these species. The host organisms, primarily orb-weaving spiders, adapted to subterranean habitats, typically prefer to remain hidden within crevices or dark recesses. This behavior serves as a survival strategy, minimizing exposure to predators while increasing the efficiency of prey capture. Their presence in open areas, particularly in locations exposed to air currents, suggested a disruption in either neurological or physiological control. Microscopic examination and subsequent genetic analysis confirmed the presence of a parasitic fungus within the bodies of these spiders. This organism, later identified as a previously unknown species within the genus Gibellula, possesses the ability to grow internally within arthropods, extracting nutrients directly from host tissues. What distinguishes this fungus is not merely its parasitic lifestyle, but its apparent capacity to influence host behavior prior to death, ultimately increasing the likelihood of successful spore dispersal. The infection process begins when fungal spores enter the spider's body. These spores may penetrate through direct contact with the external cuticle or through respiratory openings. Once inside, the spores germinate and develop into microscopic, filamentous structures known as hyphae. These hyphae rapidly proliferate throughout internal tissues, forming an interconnected network referred to as mycelium. As the mycelium expands, the fungus begins to extract nutrients from host cells, gradually compromising physiological integrity. During this stage, the host immune system may attempt to respond to the invading organism. However, in many cases, the fungus successfully evades or suppresses these defenses. One hypothesis suggests that the fungus produces biochemical compounds capable of modulating or inhibiting immune responses, allowing it to persist and develop within the host without immediate elimination. In advanced stages of infection, noticeable behavioral changes begin to emerge. Spiders that would ordinarily remain concealed abandon their shelters and migrate toward exposed surfaces. From the perspective of host survival, this shift is maladaptive and potentially fatal. For the fungus, however, it represents a significant evolutionary advantage. Open environments, particularly those with consistent airflow, provide optimal conditions for the dispersal of fungal spores following host death. Neurobiological studies indicate that these behavioral alterations may result from the fungus affecting the host's nervous system, Although fungi lack nervous systems of their own, they are capable of producing bioactive molecules that interact with neural pathways. Research on similar parasitic organisms suggests that these compounds may alter the concentration or activity of neurotransmitters such as serotonin, dopamine, or octopamine, chemicals essential for regulating locomotion, orientation, and environmental response. Through these biochemical interactions, the host's behavioral patterns are gradually redirected in ways that align with the reproductive needs of the parasite. The spider, lacking awareness or control over this process, moves toward locations that will ultimately benefit the fungus after its death. Following the death of the host, the fungus enters its reproductive phase. Spore-producing structures begin to develop internally before emerging through the spider's external body. These structures, often visible along the abdomen or limbs, release vast quantities of spores into the surrounding environment. Air currents within the cave facilitate their distribution, increasing the probability of contact with new hosts and initiating the next cycle of infection. Host behavioral manipulation is not unique to spiders. Among insects, numerous examples of this phenomenon have been documented. Certain fungal species within the genus Ophiocordyceps, for instance, are capable of altering the behavior of ants, compelling them to climb vegetation and secure themselves to elevated surfaces in what is commonly referred to as a death grip. This behavior positions the host in an environment favorable for fungal growth and spore dispersal after death. The similarities between these cases suggest a form of convergent evolution among behavior-manipulating parasites. In each instance, the parasite has evolved mechanisms that allow it to influence host neural systems in ways that enhance its own reproductive success. While lethal to the host, this strategy provides a considerable adaptive advantage to the parasite in competitive ecological contexts. From an evolutionary biology perspective, such interactions exemplify coevolution between host and parasite species. Both organisms are subject to selective pressures, the host to develop defensive mechanisms, and the parasite to refine its invasive and manipulative capabilities. The result is an ongoing evolutionary arms race that drives increasing complexity at molecular and physiological levels. Studying these systems offers valuable insights into the functioning of nervous systems and molecular signaling pathways. Understanding how parasites manipulate host behavior may, in the future, contribute to developments in fields such as neurology or psychiatry. Additionally, examining these interactions enhances our understanding of the ecological roles of parasitic fungi. By regulating host populations, such organisms may influence the structure of biological communities, nutrient cycling, and food web dynamics. In enclosed environments like caves, where biodiversity is often limited, the ecological impact of parasitic species may be particularly pronounced. Population control of arthropods by fungal parasites could contribute to maintaining ecological balance and preventing unchecked growth of specific species. Despite recent advances, many aspects of these interactions remain poorly understood. The precise chemical compounds responsible for behavioral manipulation have yet to be fully identified. Similarly, the molecular pathways through which these substances affect host neural function are subjects of ongoing investigation. Advances in genomic sequencing, proteomics, and neuroimaging technologies have opened new avenues for exploring these questions. Through such tools, scientists can investigate host-parasite interactions at cellular and molecular scales, potentially revealing the underlying mechanisms responsible for these remarkable phenomena. Ultimately, the study of fungi capable of manipulating host behavior not only deepens our understanding of biological interactions in natural ecosystems, but also challenges traditional concepts of autonomy and biological control. These phenomena illustrate that an organism's behavior, is not always the direct result of internal decision-making processes. It may instead be influenced by external biological agents operating at microscopic scales. In such cases, the boundary between self and other becomes increasingly ambiguous. A cycle that begins in the silent darkness of caves and continues through the release of millions of airborne spores represents one of the many hidden interactions that shape life on Earth. Interactions that occur beyond perception, yet resonate throughout entire ecosystems. And that brings us to the end of today's journey into one of nature's most astonishing hidden interactions. If you found this story fascinating, don't forget to like the video and subscribe to the channel. Your support helps us continue exploring the most mysterious and complex phenomena in science and the natural world. Let me know in the comments what you think. Does this change the way you see control and behavior in nature? Thank you for watching, and I'll see you in the next exploration.